lecture, which we never did so far, but since we cannot have the lab course, I prepared a few slides to introduce you to fly genetics. Okay. First of all, when you work with flies, you should, of course, know how they look. And you should be able to distinguish males and females. So that's how they look. By the way, all the pictures are from a nice book called Atlas of Drosophila Morphology. They made really nice pictures, and so I make use of them to show them to you. Okay, here you have on the left, you have a female, on the right is a male. Unless I didn't do anything wrong, when copying them from the book to the slide, they should be actually the relative size of them should be the same as in the book, but it appears to me as if the male is a little bit too small. It's in reality really smaller than the female, so the female is larger than the male. That is true, but seems to me a little bit exaggerated here on this picture.
It's a small male.
Then you see that there is the usual distinction between head, thorax, and abdomen, like you have it for many insects. All the appendages, the wings, the legs, they come out of the thorax. Each thoracic segment has one pair of legs. The wings are attached to the second thoracic segment. The halteres, which you don't see here on this picture, they are beneath the wings. They are on the third thoracic segment. Here we have a view from a side view. And you can see here quite well that the whole body of the fly is covered with bristles. These long bristles here are called macrochaetae. Then the microchaetae, these small hairs here. Here actually you have a good view of the wings, of the haltere, I'm sorry. And then you can see as a quite obvious phenotypic difference between male and female is the different pigmentation on the abdomen. So females on the 7 abdominal segments visible here, they have a black stripe at the end of each of the tergites. So these structures here are called tergites. In the male, the 5th and the 6th tergite are completely black. So in older males, this pigmentation is well visible, and you can use it to differentiate between males and females. The males don't have a 7th abdominal segment like the female has here, but of course that's not really a characteristic which you would like to use to distinguish between males and females. There are some, what I should mention is, of course, for every of these structures here, there are names. And if you consult your black course handout on pages 6 to 8, you will see that essentially every bristle has its name. So if you want to describe a phenotype, you can do that quite well. What I forgot to say here, this here is the reason why you don't catch flies so well. These are the ocelli. These are 3. So these are the normal eyes, you know. And this structure here, Drosophila has 3 ocelli. So these are sensory organs similar to the eye. So if you want to catch them, they not only have their eyes, but they have these additional eyes on the top of their head, which of course makes them much more sensitive if you want to approach them and get rid of them. Okay, then here we have the main difference, so the sexual dimorphic phenotypes between males and females, which are most most clear and which can easily be used to distinguish them. Again, as I said, the difference in pigmentation. Here again, you have the whole tears sticking out of the third thoracic segment. Then, of course, the genitalia of males and females should be different, and they are. So over here, the male looks quite different from the female. Here is, by the way, the penis. Is a short thing here is the penis, and the anus of the male is right behind it, right here. Female, this is the anus, and here is the vaginal plate. So this can also be used as a criterion to distinguish males and females. The appearance of their genitalia are quite different. Then there is a third important difference which can be used to differentiate between males and females. This is the so-called sex comb here. The sex comb is only present in males on the first pair of legs. And you see here a zoom in on it, and you can see that the sex comb consists of about 10 to 12 of these thick bristles. And people say the male uses it to grab the female. I don't know whether that is true, but it's possible. The female doesn't have this. So this is the tip of the leg. Here, there are the 5 tarsal segments, and the sex comb is on the 5th tarsal segment of the first pair of leg. So much to distinguish between males and females. That is, of course, essential if you want to do genetics. To set up crosses, you should be able to distinguish males and females. The second thing, which I thought could be a topic for this lecture, are the genetic markers. I mentioned that the first mutation was isolated in 1910, and over the decades, many more of these markers have been isolated, and these days we can make use of the most convenient of them to help do our experiments. So again, from this atlas of fly morphology, I took some pictures and we quickly go through them just to give you the impression, or that you realize how different and useful these markers can be. We start out with the different eye color phenotypes. On top here, that is wild type red. Next to it is the white mutation, so null allele. In this case, it's not white 1, like I showed you this morning, but a deletion which is called white 1118. That was basically the isolation number, also indicating white is a recessive, and the superscript always determines the allele name. There is another white allele called white apricot. Next to it here, white apricot. That is a hypomorphic allele of white. Presumably, initially, the person who isolated it called it apricot, and then later realized that it is an allele of white, and it became white apricot. So you can see the difference between this hypomorphic white allele and the wild type white allele. The eye pigmentation differs dramatically. Over here, a last example for white, that is a transgene insert. So these are at the endogenous locus, they are mutant for white, it's white 1118, but the transgene complements, at least partially, for the eye pigmentation phenotype. And by the difference from this fly to this fly, you realize you can spot the transgene, insert, and do the next crosses. This vital allele here on the P element insert complements for the loss of white activity here at the endogenous locus. Apart from white, there are many different other eye pigmentation mutations, and even though they have essentially the same phenotype, just look here, pink and garnet or prune, the eye color is very similar. Nevertheless, these are all mutations in different genes. So I give you here the name of all the mutations: CN, Cinnabar, V, vermilion, and so on and so forth. So there is quite a variety of eye color markers, and some of them have been useful for the geneticists. There are very few dominant alleles. You see that most of these alleles are recessive. That's why because of that, they are written in with a small letter here in the beginning. And here you have moiré. That is some sort of an eye color marker, a dominant one, but it's not so easy to see. You can see in the wild type up here, there is the so-called, this dark spot here is called the pseudopupil. And in moiré, there are several of these pseudopupils. And this is a dominant marker present on the TM1 balancer chromosome, but it's not so easy to use as other dominant markers. So that's why I think not many people are using the TM1 balancer chromosome. But anyway, the last point I wanted to make here is if you compare here the light allele, there are two pictures. One says day 1, day 5. And this means that this fly here was 1 day old. This fly here was 5 days old. Pigmentation is processive if you have this loss of function allele. So pigmentation can develop over time. This is also true for a white transgene insertion. This orange, kind of dark yellow, can change into orange over time. So if you want to be sure what you compare, you have to compare flies of the same age if you want to compare eye color alleles. Okay, so much for the eye color. Then we have eye shape markers. I mentioned to you eyeless this morning, this spectacular experiment where ectopic eyes could be produced. This is a dominant allele of eyeless, even though eyeless normally is, also the first alleles which were discovered were recessive. This is, the dominance of this allele is indicated by a capital D, eyeless dominant, so it's heterozygous, and you get a range of phenotype, so meaning that even though you have the same mutation, the phenotypic strength, or as the geneticist says, the penetrance of the phenotype can be variable. So you see in this fly here, about 2/3 of the eye are missing. Here, only a few ommatidia are left, and here basically the whole eye is gone. Then you see that there are other eye color mutation. maybe what I would like to emphasize again is the bar mutation here, which is present, which is the dominant marker on most X chromosomal balancers. So the FM series bar is always a good one to use. You can see that there is a clear difference between heterozygous and homozygous bar females. In the heterozygous bar, the eye is kind of kidney shaped. You just have this little indentation here in the anterior part of the eye. In the homozygous situation, where there is no wild type copy of the bar gene, you have a dramatic change in the eye phenotype. The hemizygous male here has an eye phenotype reminiscent of the homozygous female, which makes sense because here there is also no wild type copy around. This male here is bar over Y chromosome. Bar is on the X chromosome, also on the X chromosome, on the sex chromosome. Okay, that's so much for the eye markers. Here, some of them are regularly used to trace chromosomes in crosses, like irregular facets here, or glazed or drop. So these are all mutations in different genes, giving rise to relatively similar phenotypes. Then bristle and body color markers. Maybe we start out with the body color. Up here, the wild type situation, brownish, yellowish color of the body. Then there is the mutation yellow. You can see that pigmentation is much lighter. So that's gene yellow on the X chromosome, not far from white. If it's mutated, you lose body pigmentation. The other thing can also happen. Here, in the case of the ebony sooty allele, here the body pigmentation gets clearly darker, and both yellow and ebony are recessive markers and are quite frequently used in fly genetics. Then you probably look at these bristles and think, yeah, come on, there is not so much of a difference between them. That's true. Maybe again, the most spectacular is double. Here you can see that the macrochaetae on the thorax are clearly different from the wild type, and that's why stubble is a marker which people have been using frequently. It's frequently present on third chromosomal balancers because it's a very good marker and easy to spot. You will learn something about forked. Gustavo's experiment tomorrow makes use of the forked marker, and you can see that these macrochaetae, okay, here sometimes are not nicely long and thin, but are kinked at one place here. You can see it's kind of bent here also. So the bristles are have a particular phenotype which will be used in Gustavo's experiment tomorrow. Then cinched is also quite clear. You can see the hairs, these macrochaetae look like they would have been burned by fire, so they are cinched. That gave the name for this particular mutation here. Okay, then scutoid is another one which we frequently use. It lacks the four macrochaetae here on the scutellum. This trapezoid structure here is called the scutellum, and the four bristles on the scutellum, the scutellars they are called, they are missing. You see, the scutellum does not contain any macrochaetae. Yes, so you see again many different phenotypes are possible, and again, these are only a subset, and some of them are great to use in fly genetics. Then, as a final one, the wing shape markers. Up here, we have the wild type wing. You will have a chance to look at them on Wednesday morning when Shinya is presenting his experiment. Then there again are recessive and dominant phenotypes, the markers. Sorry, for example, vestigial here, very spectacular. You have only a small remnant of the wing, a vestige basically. That's why it's called vestigial. Then you have some which are more not so easy to spot. Let's say this cubitus interruptus here, that's the cubital vein. And here it is interrupted, giving the name to the particular gene called cubitus interruptus. Maybe Marcus will mention this gene tomorrow in his lectures when he introduces hedgehog signaling, I don't know. Then we have Lyra. I guess the person, it's like the instrument. He thought that this wing phenotype is reminiscent of the instrument. Then we have serrate, which is also often present on the third chromosomal balancers. And then, of course, I should mention this one here, Apterus xasta, because we were working on that together with Dimi Bieli and others a few years ago and published a paper on it. It has a very spectacular loss of wing material. Here on the distal side, you see, we also refer to it as the mitten phenotype, like a mitten you are wearing in winter, it looks like. So we can quite well explain why this part of the wing is missing, but we do not have time to.
Why? Because you want.
Hello. Who is interfering here? Okay.
Yeah, gotcha. Sure. I've termed them today. Okay.
Traders open.
Yeah. What else is pretty?
Okay.
Look at this.
Could you be quiet, please? Thank you. Okay, now it's about sex in flies. What should you know to set up a proper cross? Because in the case of these fly crosses, the females should be virgin. And I briefly want to explain to you why that it is like that. And that is explained to be on the next slide here. When males and females in flies copulate, the male sperm is stored in two organs, here in the spermatheca and in the receptaculum seminis. So over here you have the testis, and you can see up here are the stem cells for the production of the sperm, and you can see when we walk down here this tube, the sperm become more and more mature, and they have the long sperm tails. Down here are the mature sperm, which are then passed on to the female, and these sperm are stored in these two organs here. So there is, of course, not just one sperm, but probably hundreds or maybe thousands of sperm. I don't know if there anybody has ever counted them, could be. Whenever an egg, a mature egg that the female produces here in the ovaries, is passed down here into the uterus, it is fertilized by a sperm coming out of one of these two containers. And so the female can fertilize many eggs with a single dose of sperm from a male. So this complicates things, of course. If you have a female which is not virgin, which stores the sperm of a different genotype from a male with a different genotype and not the sperm of the male you wanted to use, you will get the mix of progeny. And you don't want that. This complicates the genetic analysis. So it's much better if you make sure that the female is a virgin. The other thing which one can maybe add here is upon fertilization by the male not only injects the sperm, but also a short peptide called sex peptide. And this sex peptide somehow makes the female no longer receptive to the advances of other males. So for 2 to 3 days, the female refuses to mate with other males. In this way, the male who has copulated before wants to make sure that its genes are passed on to the next generation and not competed out by another male. So these are the reasons why it is better to use virgins rather than already fertilized females to set up a cross. So then, of course, the next question is, how do you select virgins? And that is quite easy. The younger the fly, the higher the chance that it is a virgin. So that's what it is written here, how to recognize virgins, select young flies. Young flies are much less pigmented than older flies. So here are shown two males, an older male with the full pigmentation. This is a male which is less than 2 hours old, and you can see that the pigmentation of the body is clearly less than in older males. The same is also true for females. In addition, if you are collecting virgins or males and you want to be sure that they are young, you look that pigmentation is not there yet. If they are very young, like shown in this picture here, the wings are not unfolded yet. So in the pupal cage where the larva transforms into the adult, there is no space for the wings to be already unfolded, so they are packed tightly, and only after about half an hour these flies unfold and acquire the form, the shape that they have then during the rest of the life of the fly. Another sign which can help you, let's say the fly is already 3 hours old, there is another phenotype which helps you to decide. So here, this dark spot that you see here is the meconium, which is basically the first, when the fly poos for the first time, it will get rid of the waste products accumulated during pupal stage. So the waste products of pupal metabolism, they are dark, greenish, and as long as you can see this dark spot here on the side, then you can also assume that the female is still virgin. So basically what I do is I collect virgins every 2 to 3 hours. And in this way, you can be pretty sure that the flies, the females in particular, are virgin. The males, of course, don't need to be virgin. They are passing on the sperm. They can do that with many females. So that's not a problem. But the females, they better be virgin. Okay, we already come to the last part, which is how to design a crossing scheme. And we do it a little bit more careful or step by step here, because we have learned that the genetics education of few students can be quite different. Some say, come on, what you present here as genetics, that's easy. We had that at gymnasium already. We know everything. And there are other students who say, come, I have no clue what you're talking about. And so to bring everybody to about the same level, I go through step by step, and hopefully in about 10 minutes, everybody will know how to note a crossing scheme properly. So whenever you set out to design a cross, the art of designing a good cross is to cross the flies in such a way that the flies that you want are phenotypically unique and unambiguously recognizable among all the other progeny. That's the strength of fly genetics. You set up the cross in such a way that there is exactly the outcome that you planned, and which allows you to select the flies that you wanted to select. If this is not the case, you better think of a different strategy. In the handout, I have written all the rules which should be followed. Crossing schemes are easy to note down and grasp if a few simple rules are followed. So I will, of course, not continue and read this all, but we will go through all them one by one here. Here we have a female. It has on the X chromosome, so the flies have, as you remember from this morning, they have 4 chromosomes. Here, the first is always the sex chromosome, second, third, fourth, these are the autosomes. And over here we have the genotype of the male, first chromosome, so XY, in contrast to XX in the female, second chromosome, third chromosome, fourth chromosome. So if you note down the genotypes like this here, the first thing to do is only consider the chromosomes that you want to. And here I want to just point out that these flies here, they have yellow eyes because they have this P element transgene marked with white. And the males, they have wild type red eyes. You can see it has a wild type copy of white, hemizygous, because it's XY. In addition, it has 2 dominant markers on the second chromosome. One chromosome carries sternoplural. It's not important how it looks like. It's a dominant allele, which can be readily recognized. And this is the balancer chromosome for the second chromosome, is marked with curly. So it's the dominant marker is curly. So if would be stupid to take all the chromosomes along in the cross, just consider only those which are important for the outcome of your genetic cross. So to follow the chromosomes, the X chromosomes are in green, and the one on the on the second are in red. The third and fourth are missing because they don't influence the phenotype. Also, note that if both chromosomes are identical, you don't have to write both, but just write one of them. That's also a simplification to note the crossing scheme. So this means homozygous for a white minus allele. Another important rule is keep the order of chromosomes always the same. So this means always first the second chromosome, and then the second, and then the third, and then the fourth. If only the second and the third are important, write first the second and then the third. Never turn things around like down here. That can be confusing and lead to mistakes, so don't do that. Now, the next step. We have now our genetic cross. We have the females, the mothers, and here the males, the fathers, which are put together in the same vial, and they will produce progeny. So what are the genotypes of the eggs that the female is producing during meiosis? I'm sure most of you can easily tell that the female can only produce two types of gametes. Namely, either they carry the transgene or they do not carry the transgene. So this is a simple situation. A little bit more complicated in the male. There we have 4 different types of sperm. We have white plus sternoplural, white plus curly, bi sternoplural, bi curly. So these are the haploid gametes being produced in this cross here, and now we have to combine them in all possible combinations. And I urge you to use for this Punnett square here. So it's a simple matrix where on one side you have the genotype of the haploid female gametes, so white minus transgene, white minus no transgene. And on this side here, you fill in all the meiotic products for which are produced in the male testis. And then you fill in this Punnett square by combining the female X with the male X, the female second chromosome with the male second chromosome. You do that for all 8 possibilities, and this easily gives you all the 8 genotypes resulting from this cross. So this is, of course, not the end of it yet, but we are close to the end, because now you have to identify the phenotypes. So there will be differences because sternoplural and curly, for example, have segregated from each other, so they end up in different animals. Here, something I wanted to say is, this is pure Mendelian genetics, and you get all the different genotypes with the same probability, but whether these genotypes survive, that is, of course, a different question. So some markers, when they get together, the viability is low, or the genotype might also lead to the lethality. Let's say if you would have 2 curly balancers in the cross, if also the female would have had the curly over curly would be lethal. So you would never see the phenotypic outcome of that particular genotype. So that's why I write here whether they all survive and how well they survive is a separate matter. The numbers are not always like in Mendel 1 to 2 to 1, but numbers can be quite influenced by the viability of the different markers you are using. Some combinations do not survive well, and you can sometimes only get very few of those flies or not at all. Okay, so here are now the phenotypes which you have to conclude or deduce from the genotype that you have. For example, here, white minus over white plus. White plus is dominant over white minus, and you see these flies have red eyes, even though they have this transgene here. This transgene becomes invisible. It's masked by the much higher activity of the white plus copy here on the X chromosome. But what is visible here is sternoplural, so they are red-eyed and have the sternoplural phenotype. It is a female because it has 2 X chromosomes. If we look at this class here, it must be male because they have 1 X and 1 Y chromosome. And because we are now in a white minus background, you can see that we have now yellow eye color. That is the activity of the white here on the P element insertion. And we see sternoplural here. All females have red eyes because they are all heterozygous for white on the X. So eye color cannot be distinguished. That's different in the male, where one class of male has white eyes over here because they are white minus over Y, whereas one class, the other is also white minus over Y, but these have the transgene and these do not. But they can distinguish based on the dominant phenotype, either sternoplural or curly. In this way, you can see that a unique phenotype can be correlated with a particular genotype, and this is how you then select your the flies that you want to continue to cross. Let's say in this case, you could decide I want to make a balanced stock for this particular insert here. You would select these males because you know they are white minus here. They have the transgene because they have yellow eyes, and they have the second chromosome balancer because they have curly. You could select these males here and cross them again with another second chromosome balancer, basically, to make a balanced stock. Anyway, that was already all I wanted to tell you in this third lecture of the day. Now I'm luckily done, more or less, except for the last slide. You have seen that on page 16 to 18 of your block course handout, there are 4 genetic problems which you should try to solve. They are simple, nothing special. So it's with the information you have obtained today, you should be able to solve them. Then on Wednesday afternoon, when you are done with your tutoring for the presentation, you can join in on a tutorial on these fly genetic questions. I will be online between 3:30 and 6 o'clock on that day. So you can come in and ask questions related to these genetic questions. You can also ask questions about this morning's lectures. Can imagine that not everything was clear. I would like to remind you that there are also a summary, basically, of the lectures that I presented. It's in written form. It's, what is it called? Genetics handout or something like that. And there is another written summary of fly genetics by Michel Markstein. I encourage you to read those. They should help you to figure out what I told you this morning. Then on Friday afternoon, following your paper presentations, we will in detail discuss these 4 genetic problems, and at the end of the day, everybody should know how to properly write down a crossing scheme. So thanks for your attention, and now I think you continue with reading papers and joining later your tutors to discuss first questions you have from your first reading of this paper. So thanks for your attention, and I wish you a good continuation of the course. See you.
Thank you, Martin. Can I maybe just say one more thing before everybody leaves? So now is paper reading. First glance at this paper. Then you discuss with the tutors, and tomorrow during my lecture, the lecture will be on signaling, but the course afterwards from 10 to 12 will be on signaling and how you analyze it using genetics. So I'll go back and use what Martin has explained you now, again, by using a whiteboard to show up a few things. This is a P element, it's on this chromosome over a balancer. So you will hear the things Martin said. You'll use them tomorrow, or you see useful examples as well. So that will help you to understand it much better. And maybe on another note, I would very much like if tomorrow, not during the lecture from 9 to 10, but during the lab from, I don't know, 10:30 to 12 or so, it would be nice if all of you could turn on the camera at that time, because it should be an interactive lab, kind of. You look at pictures and then we'll discuss it. So I just wanted to warn you a bit. It would be nice if many of you turn on the camera. So if you don't want to be on the pajamas on the camera, then it would be nice. Then you know what will happen at 10:30. It would be nice that many of you would watch, and then we can discuss these slides a bit together as we do in a practical course. And then it's nicer to see the people tomorrow. During the lecture, I think it's okay. But then in the real lab, it would be actually quite nice. Then we get to know each other a bit better as well. Okay, so paper reading and then first tutorial. And thanks a lot, Martin.
Kein Problem.
Tschüss zusammen.